Hi, and thanks for joining us today. So today we're going to talk about ways to use an NCBI account in a traditional capacity as a professor or an instructor in a college, a university, or even as an AP biology teacher. My name is Rana Morris. Sherry Holland Bailey is here also. I've been in the professor's shoes, so I have an idea as to what you guys do. Sherry is an expert in the NCBI account system in My NCBI, so she'll have all the good technical information if you have specific questions. So professors, you're busy, really busy. In your capacity, you have to develop and teach your courses and labs, coordinate and run your journal clubs and seminars, direct your lab's research efforts, write grants and publications, counsel and mentor your students. And while doing this, stay current on everything related to both teaching and research topics. NCBI has information that would help most of these efforts, but there are so many interesting records and so little time to organize them for efficient use. Sign up for a free NCBI account, or if you already have one, we're going to show you some ideas as to how to organize your important lists and use them in what you're already doing. This webinar will show you how to use the NCBI account and give you just some ideas that I've thought up. You'll see how to save searches and get automated updates when new publications or database records are available, maintain custom collections of important records for use in coursework and research projects, create lists of publications or database records to send to your courses, journal clubs, and even research teams, and maintain your bibliography, create a URL to share, and often you need to post your listing of publications on your faculty profile page. We'll show you how to get a URL to put on your faculty profile or web page. And finally, you can reuse some of this information to very quickly create a federal government BioSketch that you can use in your granting, all with a click of a mouse. And you can keep track of everything right on your My NCBI dashboard. So first, you need to sign up for an NCBI account. The steps are outlined here, and there's lots of information again in the information provided to you as to how to do this. At the header of virtually all of the NCBI pages is a link that says sign into the NCBI. If you click there, you'll get to a page that looks like this that has a way to sign in either directly to NCBI or using a Google account. At the bottom is a link to register for an NCBI account. So you can click this button down here to set up your registration, and if you do, you'll actually see a screen that says success, yay, you are signed in. To verify your email address, to make sure that this really is you, we'll send you an email. It looks like this and has a link to validate or activate your NCBI account, and then you'll log in and see that your email address has been confirmed. Again, the registration page has a direct URL, which is listed here, and we actually have an NCBI help book that has step-by-step instructions. Or if you already have an NCBI account, you can sign in, and you can actually sign in directly here, which I'll show you in a little bit, or you can sign in with your Google, NIH login, ERA Commons, or other account. And again, the direct sign-in link, and there is a step-by-step help book, just in case you need it. Now, once you're logged in, you'll notice again at the top header of most of the NCBI pages, there are 3 links here. One of them is your username. In my case, I'm an NCBI professor. And that will actually take you over to a page that will allow you to take a look at your account settings. Here, you can actually change your password if you need to. You can change your security question. You can add additional accounts, such as ERA Commons, in case you're attempting to get an NIH grant. And there are other things you can do as well based on your account. That's your username. If you click on My NCBI, it'll actually take you to a dashboard view. And this is really what we're going to look at today, because this particular dashboard view is helpful to keep track of all of your various lists and collections. The nice thing is that the NCBI account does have a help book on the NCBI bookshelf that really has step-by-step instructions of virtually everything you think or you didn't even know you needed to do. On the My NCBI dashboard, you can customize this page. You can actually reorganize or get rid of or move these portlets around, these widgets around to something that's helpful for you. There is a video overview that we've created that will literally step you through the entire process as to how to make this page look exactly as you would like it to look. All right, so now what can you do with this? As a professor, you probably already search lots of databases. PubMed, you could search Gene, you could search MedGen, even PubChem. Often you want to find and collect materials for teaching. You may want to keep up with your research competitors and what's going on in your research field. Check on your own publications. Are they listed in PubMed? Do you have one in PMC yet? Often you might, I have often been told about a relative's diagnosis and they want me to check out information because they think I can find the information for you. You can actually, you need to routinely search various databases, and I'm sure if you could, you'd love to be notified when new records are available. You can do this in the Saved Searches widget on your My NCBI dashboard. These are two that are there currently on the NCBI Professor dashboard. Malaria and lactate dehydrogenase is a research project, and, well, my father's age-related macular degeneration file as well. Often you need to create collections of materials. Maybe you've got printed lists of references sitting on your desk, or a digital list saving on your office computer's desktop somewhere. Or maybe you've got a wall filled with sticky notes or a drawer full of bar napkins. That's how I used to collect them. It'd be really great to find a way to organize custom sets of records and be able to access them from anywhere, from your office computer, on your phone if you're in a meeting, at home in the middle of the night when you're actually doing some additional work. You can do all of this by saving your lists in your collections widget, which again is on the dashboard. And these, again, are some examples of things that I've set up as a professor. Maybe a freshman seminar on malaria and the international implications, an LDH biochemistry lab folder, and even, again, mom, maybe dad's macular degeneration. Now, once you have all of these lists and you've created these collections, you often need to share them with students, colleagues, or administrators. There's a lot of reasons you might want to share a list. Maybe reading reference lists for courses or labs, or journal clubs or seminars. Maybe, again, you want to have that link on your faculty profile page of all of your publications. Maybe you need to share them with the administration so they can see your productivity. Yeah, you can share these collections too, and we'll show you how. So this is just a quick overview, and again, I'm not going to go through too many details because I'm going to show you in a demonstration in just a little bit on the live side if it lets me. Once you've actually done an interesting search, you can save it. Underneath the text box is a Create Alert button that will take you to a page that will allow you to save a search and simultaneously set up an alert system that will send you an email when there are new records. And I'll show you how to do that. Again, there's a great webinar video that'll actually show you how to do this, or there's step-by-step instructions in that help book. You can create collections online, which you can access anywhere. As an example, let's say you do a search in PubMed and you find a really great bunch of records. You want to create a brand new collection with that, you can just click the Send To link in the upper link, Collections, Add to Collections, and you can either create a new one or append to an existing collection. That works if you're on the results summary list. It also will work if you happen to be on a particular record page where you can send that record either to a new collection or append to an existing collection. And it's not just PubMed, it's also Gene and many of the other databases as well. And again, step-by-step instructions in the NCBI account help book. Finally, for now, it'd be really, really helpful to make available or share these lists of references with students, colleagues, and administrators. You can set up specific collection folders just to do that. Once you've identified a particular collection, such as the LDH lab collection that you want to share, you need to change the default designation over here. Private means you're the only one who can access it. That's the default. If you change that to public, it will actually give you a URL after you save it that you can post and share with people, and then people will be able to see the contents of that particular collection. I'm sure this is a bit complicated, but it really isn't too bad, actually. Let me demo it for you live. So this is what I'm going to do. I'm going to sign into an NCBI account. We're going to do a search. We're going to save the search and create an alert. We're going to create a collection. We're going to save a record to that existing collection, and then we're going to make that collection public and share the URL. For this demo, I'm going to find some references to start a student off with a senior project. She'll be researching malarial diagnostics, focusing in on the Plasmodium falciparum's lactate dehydrogenase enzyme as a target for detection and eventually drug design. So the first thing I want to do is sign in to NCBI so that I can start a collection that I can share with her. So this is the NCBI homepage. Let me show you how to sign in to NCBI. In the upper right-hand corner is that hyperlink to sign into NCBI. And when you click it, it will open up this page. Now, I already have an NCBI account, so I'm just going to sign in directly here. NCBI Professor. Now, let's see if I can remember my password. And it is still spinning in the upper corner. Aha. I'm logged in, and I know I'm logged in because now in the upper right-hand corner, it doesn't say sign in. It says NCBI Professor. If I click here, it will allow me to go into the account settings, change my password if I need to. The My NCBI link will allow me to go to the dashboard, and we're going to take a look at that dashboard. And these are lots of widgets. There are all sorts of widgets, things like Save Searches, Collections, My Bibliography, Science CV. There is even a Blast History widget, and you can click and drag and move things around if you'd like to, or even get rid of them. Again, there's a video overview that will show you an actual video of how to do this, and there's also the help link as well, which goes directly to that help book. So first, I'm going to go back to the NCBI homepage. I'm going to click the double helix NCBI to do that. And I'm going to start a search in PubMed to find up a couple of publications I think I'd like the student to be able to read. So what we're going to do is we're going to research articles about malaria and diagnostic. I'm going to go to PubMed, and we're going to search for malaria and diagnostic and the particular enzyme I want her to concentrate on is lactate dehydrogenase. I'll run the search, and we'll pull up a number of publications that are related to this. Now, this is a senior student, and so what I want to do is just give her a few review articles for her to look at. So on the left-hand side of the screen, I'm going to click Review. And to make it easier for her also, we're going to go ahead and click those that are just free full text. Because of that, we can pull some that happen to be in the PMC database, and you can see that here under free PMC article, and she can actually access the full text article anywhere she is. So I'm going to pick a few of them that are interesting. One of them is here, recent progress in the development of diagnostic tests for malaria. Here's another good one, biomarkers and their applications for rapid and reliable detection of malaria, and it specifically talks about lactate dehydrogenase. And the fourth one, why not? Malaria rapid diagnostic tests in endemic settings. Those are three particularly good articles, and we'll go ahead and actually save those to a collection. So what we're going to do is click Send to, Collections, and there are 3 items here that I've selected, so I'm going to add to collections. Now, if I hadn't checked any of those boxes, it would have sent everything. I know it's not obvious, but that actually works very, very well if you're trying to do a large set of appendings. So we're going to go ahead and create a brand new collection for this. This is directly for the student, and we'll call this, oh, I don't know, malarial diagnostics, and click Save. And this is going to go ahead and create a new collection for me. And in fact, it's going to show it right here. So I'm going to open up the dashboard in a new tab so you can take a look and make sure that that collection is actually created and added onto my dashboard. Here's the collections, and there it is right at the bottom, malarial diagnostics, and there are 3 articles in there. Now I'm going to go back real quick to the original search. And remember I said it would be awfully nice if it would just let me know, this is an interesting topic to me, would it be great if it could actually give me an alert and let me know when there are new articles. So I'm going to go ahead and underneath this say Create Alert. And this is going to do two things at once. One of them is it's actually going to create a saved search for me. I can just rerun the search again if I want. But at the same time, since I clicked Create Alert, I'm going to go ahead and say, yeah, I would like to have email updates of new search results. So yes, please, email them to me, or I could have them emailed directly to the student if I wanted. The schedule, monthly. I could make it more often than that, but for now we'll just stick with monthly, the first Sunday of the month. Give me a summary report format. Don't show me all the abstracts. I'll go there when I'm interested. And send at most 5 items. I can do more if I want. I'm going to stick with 5. And then I'm going to click this checkbox. And I like to do that, send even when there aren't new results, just to make sure that I know it's actually working. The nice thing is that not only is this search actually the one that they're going to redo, but it also includes the filters, review, and free full text. So all of that is already included for you. I'm going to click Save, and then I'm going to take a look back at our dashboard again, and I'm going to reload the dashboard, and we'll take a look and make sure that that search was actually added to my dashboard. And here it is, malaria and diagnostic and lactate dehydrogenase. When was the last search? Today. Now, as an example, the age-related macular degeneration one that I already had, and it was searched 4 days ago because that's when I created it. Since that initial search, there are 22 new articles that have come out with that particular search. So I now have an email alert for when, monthly, sending to me about malaria diagnostic and lactate dehydrogenase. I have a really great collection, beginning collection, with 3 PubMed articles on it. But let's add one more thing to that. It doesn't just have to be literature. It can be something else as well. Since I'm interested in having her actually do research on the malarial lactate dehydrogenase enzyme, let's go ahead and add the gene record for the malarial lactate dehydrogenase enzyme. And the malarial organism is called Plasmodium falciparum. So we're going to look specifically for a gene record that will have information about that particular enzyme. Our gene database, if you're not familiar with it, is a summary site that has a link to everything we know about that particular gene. Lactate dehydrogenase, there are a couple of enzymes that actually have that activity. The classic one is the one at the top. That's this one here, Plasmodium falciparum, L-lactate dehydrogenase. So we're going to go ahead and actually add that to the same collection. So we're going to create a mixed collection of those 3 publications we just found, and we're also going to add this gene record. So I'm going to go ahead and click the Send To link. Got a new laptop. I'm not quite sure why it's doing this. Then I'm going to click Add to Collections, and it's going to say, do you want to start a new collection or you want it to append it to an existing collection? And I want to go to an existing collection. I want to go to that malarial diagnostics collection. I'm going to hit Save and allow it to append that gene record directly to that existing collection. New items were added to your collection. So I'm going to come back to the NCBI, My NCBI dashboard. Here's the malarial diagnostics that originally had 3. I'm just reloading the page. And now we have 4. Now, if I want to make sure the student has access to this particular collection, right now it's private. I'm the only one who can see it. If I click the private link, I can actually change that to public, from private to public. And when I do that and save it, it will allow me to have an actual URL that I can then share with the student. And here's the URL right here. So that's the URL I can share with the student. And this is what the student will see, My NCBI collections, and here are those 4 records, here are the publications, and here's that gene record. Now again, there are YouTube videos, and there are also step-by-step instructions as to how to do just exactly what I've shown you how to do. So if you have any questions, there are plenty of ways for you to, depending upon whether you like videos or whether you like step-by-step instructions. Another common thing that you often need to do as a professor is to maintain a bibliography listing, a publication listing for various purposes, whether it's populating the publication section of for your CV, the department or school admin's annual report you have to file, or even just to list your productivity on your university faculty page. Your NCBI dashboard has a widget called My Bibliography. Now, My Bibliography is actually just a specialty collection. In fact, you can see it listed in the collections. This collection is for PubMed records or PNC listed publications, but you can also add non-NCBI records to it, such as non-PubMed journals. So if you're a chemistry professor and you have a publication in a chemistry journal that's not listed in PubMed, you can actually manually type it in. Textbook chapters or book chapters that you've actually written, you can include there as well. You can add those to the My Bibliography collection. Just as for other collections, once you are happy with it, you can make it public and use the URL produced to share or even post on your faculty page. How do you do that? Click Manage My Bibliography, and you've seen a page that sort of looks like this before. At the top is a link right here where you can make your bibliography. It's currently private. You can make it public. And again, use that URL just like I showed you before. And here is the public listing of your publications. Then you can add that directly to your faculty profile page or your web page, and people will automatically see the up-to-date list. If you do it this way, you only need to maintain your My Bibliography listing, add new publications as they come out, and whoever has this URL link will always have the up-to-date list without you having to do any extra work. Additionally, you can use My Bibliography listing to export the whole list to a citation manager for adding references for your next publication. And you can also use this listing to make a BioSketch to add to your federal grant proposal without having to do too much extra work. A lot of the instructions about My Bibliography are available on the help book and also the step-by-step instructions to share your bibliography. Now, preparing a federal grant proposal is hard enough. Often it gets pretty tedious. NCBI has worked as a team with a number of other organizations and federal agencies to create a tool to help you to simplify the BioSketch generation process. The dashboard widget is called ScienceCV. You can actually see it down here at the bottom underneath My NCBI. It's designed to repurpose the information that you've already listed in your My Bibliography, along with stored information that you put into your NCBI account and, for NIH grants, your ERA Commons account. It takes this information and populates and automatically formats your NIH or NSF BioSketch. So again, My Bibliography is a normal collection, and ScienceCV uses that information. You can manage ScienceCV by creating a new BioSketch, and you can select, do I want an NIH BioSketch or an NSF BioSketch, and there are other agencies and organizations that are coming online in the future. You can choose your data source, whether it's an existing BioSketch or some additional things such as ERA Commons or even OrcID. And then you can create your BioSketch, and you can export it either as a Word doc or as a PDF. So here's an example of an NSF BioSketch and an NIH BioSketch. Once you do get your NIH grant, congratulations. Your My Bibliography will help you monitor your compliance with the required NIH public access policy. And it will also assist you with the preparation of your annual and final reports because you have your publication listing at NCBI. We won't get into that right now at this webinar. We've produced others that talk extensively about this, and there are many documents at NIH and NCBI pages that will help you with this. And the handout that you were given, and that will also be in the blog, will also have links to managing NIH public access compliance. I want to warn you, or perhaps alert you to, a couple of new developments. NCBI has slowly and recently been shifting over to a new design principle, user-centered design. This means that we'll be updating our resources and web pages with more modern and usable layouts based both on what users need to do and also rounds of testing with real people even before we release them. This may impact you in the near future in two specific ways. Coming in the fall of 2018, there will be a new look for the My Bibliography widget. This is how it currently looks, and here is a preliminary design of what it might look like when it's released this fall. We've done this to fix issues and solve concerns that many customers have expressed to us, and we've also come up with a few of our own ideas about how we could really make things better and easier to do. We're currently in rounds of testing right now, but one thing I want to make sure you know, everything that you do now will already be included in the new system and be fully available in the system. So you don't need to wait until this comes out. Start getting used to the system, put your information in, and it will automatically be converted over. And we hope the new pages will be a whole lot even easier to use than they currently are. And as it gets closer to the release of the new system this fall, we'll do another webinar to show you exactly what it looks like and then how it's going to work. Another announcement is that coming in spring of 2019, there will be a new look for PubMed. It's been a while since PubMed has been redesigned. This is what PubMed looks like now, as I'm sure you know. In this recent version, as mobile devices have become more prevalent, there's been an attempt to make a mobile version, which works pretty well, but doesn't have all the full features, including a lot of the My NCBI features. We'd like to make this user interface more modern, easier to use, and better on all screens, regardless of size. For the past several months, we've been running an experiment called PubMed Labs. If you take a look at the PubMed Labs, you may see what the new version of PubMed might be like. I'll show you that in a second. But please know that we'll have a lot of announcements and webinars, not only letting you know when this is going to happen, but also showing you how to use the new pages. So to get a clue as to kind of the direction that we're going, you can try out PubMed Labs. The URL for PubMed Labs is at the top of the page. This is an online, fully functional sandbox for testing out layouts, features, and functions with the full set of PubMed data. You won't actually miss any of the PubMed data. It's all included in PubMed Labs. People have expressed to us already that it seems to be easier to use and easier to read, and we've been able to add new functionality that many people have been asking for. It's pretty nice for large screens of computer desktops, but it also seems to work fairly well on mobile devices as well. Please keep in mind that PubMed Labs is not yet fully compatible with your NCBI account and My NCBI dashboard, but before it's actually released as PubMed and released to the wild, it definitely will include all of the My NCBI features. I've gone over a lot of stuff, and there's always more things happening at NCBI. If you want to keep up with what's happening, including information about new developments, upcoming webinars, when we put out cool new YouTube tutorials, you can scroll to the bottom of any of your web pages and click on any of the social media links. Or you can watch a webinar that we did on keeping up with the NCBI that's on YouTube right now. Finally, if you ever have any questions or need help with NCBI resources, please don't hesitate to send an email to info@ncbi.nlm.nih.gov and we'll get right back to you. Thank you very much, and Sherry and I will be very happy to attempt to answer any questions you might have.
Thanks, Rana. We're at time, but we'll keep it open and answer some of these questions. First one I'll answer because I think I understand the answer to this question, and then I'll have Sherry and Rana talk about some of the other ones. One question was how public is the public URL that you create for the collection? And the answer is that URL is, whoever wants to see that is going to have the URL. It's not indexed by a search engine, so nobody should be able to find that. The second question is one that we can talk a little bit about the pros and cons of these two approaches. The question is, do you recommend the saved search feature or collection feature for sharing a systematic review list of resources? And I will let Sherry address that one.
Great question, Brian. The answer to that question is it depends on what you want to do. For the saved search, it's going to give you everything that's indexed based on your search. So that means that something could have been written, for example, in 2007. If that is not in your search, but it was indexed recently, it's going to show up in that emailed saved search. If that's all you care about are things relating to that topic, it is perfect for you. For a collection, these are things that you want to save and refer back to later. So honestly, the best answer for this question is you can use them in tandem with each other. You can use the Save Search feature to pull all that documentation for you, all of those citations, and then the ones that you would that you want to keep and perhaps even share, you would put into the collection itself.
Okay, thanks, Sherry. And I think Sherry addressed the second part of the question, which was about the search strategy, which will be present in the email, but not in the collection itself. Somebody encountered a problem. Somebody said that if you have a mixed collection, for example, with gene results and PubMed results, that a patron would not be able to open the collection. And I don't know that that's true, but I'll let Sherry talk about that.
They've been making changes to how collections work. For the particular example that Rana used, she used a gene as well as a PubMed. And you can try this out in your own account as well. And I just tried it out here. It does work. It depends on what you're trying to do with that collection. If you're just going through and sharing this information, it's fine. If you're taking that information and trying to incorporate it into another collection, that's where you're going to run into a little bit of a problem, but we're working on that as well.
Okay, so those are the questions that we have so far. And so I'm going to go ahead and ask Rana to end the webinar. And thanks everybody for coming, and we'll talk to you next time.